Okay, good morning. Let's have a class. And we will move on to exam 1, lecture 3, which is about overview of microorganisms' cell structure. And basically, we're going to focus on the bacteria. This will be lots of information, and some of them we will talk in more detail later on. So first of all, there is a comparison between prokaryotes and the eukaryotes, the cells. And I want to make sure you understanding this. You already know what is the difference between that. It's called cell membrane binding system. So you learned in your biology class already. Prokaryotes, they don't have a cell membrane system, so everything stuck together. And the eukaryotes, they have an individual cell machinery, and they are separated. It's functional independent. Of course, they work together as a whole system. And there is a once very well-known theory behind it. Is called endocytosis. So this is a very important concept and theory. So they think a long time ago, there is a small microorganism, a teeny tiny live body. And they are carry on the functional, looks like a lysozyme, looks like a mitochondria, or looks like a Golgi apparatus. And then they eat it by a fungi, let's say, eukaryotes. And then the genomic information was integrated into the whole large system, and finally it's become part of the body of the eukaryotes. So very similar to like parasites become a host. So that's called endocytosis. So there is a cell machinery, let's just say that. Cell machinery. Okay, this is carry on by let's say tiny microorganism. It could be a virus even. And then what they do, this whole thing is eaten or engulfed by a fungi, let's say. Okay, this is a big one. It's a fungi, eukaryotes. And with more the evolution developments, this section becomes part of the body. So that could be, let's say, mitochondria, for example. Okay, or a Golgi apparatus, also structure. So that's called the endocytosis. Now we also want to mention that we know the bacteria, they have a toxin. They'll cause a problem. So for example, E. coli, 0157H7, they have a Shigella toxin. And this have a very similar theory, which is a long time ago. There is a bacteria phage, we call it a beta phage. And this phage carry on the Shigella toxin and integrated the DNA sequencing into the chromosome of a bacteria, which is E. coli. So later on, this has become a toxication material. And this theory, very similar to endocytosis, we call it a lysogeny conversion. So very similar, okay? They think a small body carry on some of the information. They eat it or engulfed by a big one. And later on, they stay together. So we can say this is kind of like a parasite. And the whole thing is kind of like a host. Okay? So that's very simple to understand behind it. Now, in this class, we're not going to go talk about the anything you already learned in your biology class. We will focus on the bacteria, something we have not been taught, or something talked from a different perspective. So it is very simple. If you look at the prokaryote cell structure, you have an inclusion, you have a capsule, cell wall, plasma membrane, fibril, ribosome, nucleic acids, plasmids, and the flagella. It looks like it's very simple. So I remember a long time ago, I have a question for students. I said, what is the major difference between eukaryotes and prokaryotes? Somebody, they just write one sentence said, prokaryotes looks like simple and eukaryotes looks like complicated. Yes, when you look at this information, seems like that, but think about a bacteria in their very short lifetimes. It can be functional as good as a human being even. This structure does not mean it's simple. It's more functional. You could say understand about that way. And this slide is very important because it gives you an overview of the prokaryote cell wall structure. So including cell membrane have a lots of functions. It's a communication of the nutrients, metabolic process, detection of environmental signals, and the cell wall, the shape, protection, ribosome, we'll talk about that in little detail, protein synthesis. The protein synthesis we'll talk about in the exam 3 when we talk about translation. Capsules, we already mentioned about the capsule stain. Phyli, which is attachment, conjugation, transformation. That's for bacterial genetic information, communication, and motility. Flagella, we talk about the true motility and the storage of the granule, inclusions, that's for carbon, phosphate, and others. So it's a very easy slides when we talk about the overview of prokaryote structure. So this we're going to move on to the bacterial cell wall membrane system. We talk about the plasma membrane, cell wall, and the layers outside of the cell wall. So let's talk about something detailed. Okay, this is everybody knows the fluid mosaic model to talk about double layer of cell wall membrane. And we want to, I still want to spend some time to get a little bit detail about it. So, it's very simple. We already draw the picture before. We're going to draw again right here. Okay, we have phospholipids. This is everybody knows. And lots of the information we could be talk about together. Okay, phospholipids with something in the middle that is called a protein. That's transmembrane protein. So we have this guy comes out, phospholipids, and we have this guy here is protein. Basically, those two major chemicals, they're in a movable, balanced system. That's called the fluid mosaic model. It's not stay there, not static, it's moving. And regarding the protein, there is basically two types of the protein. This is type of what we call it an integral protein. It's like a channel to transport nutrients inside of the cell wall system. Basically go here, and the waste will be transferred outside. Now, we also want to be paying much attention. There is some of the protein, even right here, attached to it. Okay, this is called peripheral protein, so which means they're on the side. Now, sometimes you will see some protein like this, like that. A little bit thin, but also transmembrane. In the middle, that could be heme, composed by different amino acids. And sometimes you also could see there is a little bit of chain like that happens. That is called oligosaccharides, okay, which is a sugar. So that's a basic structure. You already learned Freud the mosaic model. Now we want to talk about these things a little bit detailed. First of all, we want to dig a little bit more about these phospholipids. So everybody knows phospholipids. The head, hydrophilic. The tail is hydrophobic. So what means hydrophilic represents, we say it's like water. So instead, it's very simple, hydrophobic is dislike water. So this you already know. Now some people will be messed up with those. Okay, if you ask them to label hydrophilic and hydrophobic, they will be messed up. But there is a way to remember that, to understanding about that. So we need to know the structure, a little bit of chemistry, and also organic chemistry. So let's talk about organic chemistry. Why the top of the head is hydrophilic? Why the tail is hydrophobic? We could talk about from the chemistry standpoint. So what it looks like. Okay. CH2, CH2 goes here, goes phosphate, like this. Okay, then you go here, will be CH2, then you go CH, CH2, then you go here, will be O, O, C, O, and CH2, 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 and that's going to be very long. This is basically a chemistry structure of the phospholipids. So then we can talk about that a little bit. This is the area will cause hydrophilic like water. Why? This is called ethanoamine. What means ethanoamine? It's an alcohol-like chemical. And alcohol can dissolve in the water. So that's very easy to understand. Now obviously when you see this is a phosphate. So the head here is hydrophilic. It's like alcohol can dissolve in the water because of the structure. How about here? What is this part? This is glycerol. And this is called the carboxy group. And what is this here? That's fatty acids. They are not dissolving in the water. Like the oil floated on the soup. Is that right? You see those bubbles? They're not dissolving. Now what are the fatty acids? This is basically, if the model is a C, C equals 16. So in the chemistry, you know about that. If C16, 0, that's palmitic acids. If it's C double bonds, no double bonds, C18, that's stearic acids, which is we are from the beef and the pork, the major stearic acids. We don't like that. If we have a C, 1, that's 1 double bonds. Unsaturated fatty acids, that is oleic acids. How about like C18, 2, linolenic acids? Is that right? That's what you learn in the food chemistry, but in the chemistry class, organic chemistry. But those fatty acids, basically, they are not dissolved in the water. And there's only one exception for fatty acids dissolved in the water is a short-chain fatty acids. Let's say lactic acid. Let's say acetic acids. Okay, that's also fatty acids. Is that right? That's dissolved in the water because it's a very short-chain fatty acids. The long-chain fatty acids are not dissolved in water. So that's something we want to mention about. It's interesting to know about that. In the top, ethanolamine looks like alcohol products, phosphate right in the middle, and the bottom is glycerol with fatty acids, so they are not dissolved in the water. So that is a fluid mosaic model, what it looks like. And the structure looks like here. The cell membrane system, what is the function? Well, that's pretty easy. Cell wall synthesis, energy metabolism, DNA replication, we'll talk about in exam 3. The censoring of the stimuli, we talk about the chemotaxi, we'll talk very briefly, and the molecular transport. Okay, we will talk about the molecular transport real quick. in the next slides. These couple of slides, we will be put them together for you to understand, since we have a structure here. The first thing, let me just remove this. So we talk about the fluid mosaic model. We already mentioned a protein, transmembrane protein, on the cell membrane system. So automatically, we will going to talk about is nutrient and waste. I will say together, transport. Okay, but lots of the textbooks, they don't talk about this. So they become a nutrient transport. Now, nutrient transport, you already learned in your class, the biology 101 or different type of the classes, there's basically three methods. Passive diffusion. So passive diffusion, if we say it very simple, that's from higher concentration to lower concentration area and no energy cost, so no ATP used. Same as a human being and animal, bacteria use passive diffusion, but very limited, only for water, carbon dioxide, oxygen, those very simple but important chemicals. So that's passive diffusion. The second one, we call it a facilitated diffusion. Facilitated diffusion. So facilitated diffusion, very similar to passive diffusion, a little bit difference. First of all, from higher to lower concentration, and no ATP used, but needs a transporter. And we also call it a carrier. Well, the carrier basically is an enzyme, and basically it's a protein, because the major structure of enzyme is a protein. So facilitated diffusion, if you want to compare to the passive diffusion, you can draw a curve. X-axis, this is a concentration. And the y-axis, this is a rate of transport. And what it's going to be looks like, it's very interesting. This straight line is passive diffusion, and the facilitated diffusion looks like this. There is going to be a flat area, which is saturated area, because of the enzyme. Okay? But I don't want to spend too much time talking about this guy. Why? Facilitated diffusion is from higher concentration to lower concentration. For the bacteria, it is barely used, especially in the real environmental scenario. So you think about this way, if there's a bacteria on the bench is a poor nutrition area. It's not like the media we did in the lab. There is not much nutrition, so the bacteria has to survive. So obviously they don't use facilitated diffusion, because on the bench is a limited amount of the chemicals and only the water or the moisture. So we can say for bacteria, facilitated diffusion only for certain species in certain environments. Most of the time, it's not really useful. Basically, for some fungi, yeast and molds, they use facilitated diffusion. So we don't really care about this guy here. Okay, so we just make sure we skip it. Okay, next one, active transport. This is you already know. In the opposite direction, from lower concentration to higher concentration of the nutrient ingredients, they need ATP, which means they need energy, and they also need carrier. Now, for bacteria, they did use that a lot, sometimes. So what happened is, remember we draw the picture here, and I said the protein important is integral protein, we call it a transmembrane protein, and we also has a peripheral protein. So what happened? This is basically is a carrier. And the carrier is an enzyme, we call it a permease. Look at the words permease, like a permit. Will carry on something into the cell membrane system. And this one we all talk about called the solutes. Some chemicals going to go inside, okay? Now, when they go inside, they need energy. Where's the energy? The energy basically coming from peripheral protein. So in here, they're going to have ATP, hydrolyzed, become ADP, and phosphates. And here's the same thing. ATP, hydrolyzed, become ADP, and release the phosphates. Okay, so what are the basic mechanism behind? We call it the ABC transporter. What this means, which is ATP binding cassette transporter. So we call it the ABC transporter. Now, in the biology area, you also learn that the permease, the carrier, and the solutes, they could be in the same direction. So they could be go the same direction, go this way. That's called a syn-porter. If the carrier go this way and the chemical go the other way, we call it an anti-porter. Okay, these are just briefly review what you learned. So that's active transport. So we reviewed what you learned in your biology class. And I would say this, these three methods, we talk all the time in biology class, honestly for bacteria, they are responsible basically 40% of the mechanisms they use for transport bacteria, transport nutrients. For bacteria, the nutrient transport mainly rely on is group translocation. Okay, so what is group translocation? We would love to explain to you, but there is a little bit of biochemistry stuff behind it, and the biochemistry we'll talk much more detail in the exam 3. So here is the thing, you always remember, for bacteria, get nutrients. The most economic way and the most efficient way is group translocation. It is not a passive diffusion, precipitate diffusion, and active transport. Group translocation rely on a chemical called the PEP, called the PEP transferase system. What is this guy? Phosphoenolpyruvate. Now you need to know where this comes. So this goes to, we need to review glycolysis. We're going to talk about detail, but just give you some hints here. So what that? Glucose become glucose 6-phosphates, then become fructose 6-phosphates, then become 1,6-biphospho-fructose. This is a linear area, so you go to the branch. So you go glyceroldehyde 3-phosphates. Okay, 1,3-biphosphoglysarate. And you go 3-phosphoglycerate. Okay, it's too long. 2-phosphoglycerate. Then here is the important one. Phosphoenolpyruvate and go down to pyruvate. This is the key thing is right here. Okay, that's glycolysis. That's where it comes from. Now this is we'll have all the chemicals. If you don't know, you can draw a very simple curve. It looks like this. 1,6-biphosphor fructose, and then it becomes this one. Goes a branch. Go down is pyruvate, and in here phosphoenolpyruvates. Okay, that's you have to know where it is. So this is what happened. Phosphoenolpyruvates, obviously, this guy is carrying phosphates. And this phosphate is going to go into a second chemical. This is called enzyme 1, which is a chemical, has a phosphates. Then this goes the next one, which is HPR. This is called heat stable protein. And this is also going to carry our phosphates. Then they go to the cystine. The cystine actually is connected to a double layer, the fluid mosaic model of a cell membrane system. So that's what it is looks like. Then go here, you have enzyme complex 2, enzyme complex 2 right here, and also enzyme complex 2 right here. So this phosphate will be transferred, and if you have a glucose, what will happen? This glucose can go in, directly become glucose 6-phosphates. What this means, this step happens directly. Instead of like that. Did you feeling about this is more complicated? You have a protein that has to be a transmembrane protein, and you need energy. You need the carrier, and the concentration of the carrier needs to be an appropriate amount. Otherwise, it's going to be, if it's saturated, the transport speed is going to be decreased or flat. Lots of the condition for this guy. But this is easy. You have precursor phosphate enolpyruvates, and I just transport directly glucose can become glucose 6-phosphates, could go glycolysis. So makes the metabolic process very efficient and very energy economical. So that's the reason group translocation is used by a lot of the bacteria, especially like E. coli, to transfer lactose, glucose, and also mannitol, maltose, all those type of major 5-carbon, 6-carbon sugar. They're using group translocation. That's a widely used, most studied mechanism for the bacteria. Now, why we call it a group translocation? Because during this process, when they transfer phosphates, they have enzyme complex 1, they have a heat-stable protein, they have an enzyme complex 2, and we call this, looks like a group. That's why we call it group translocation. Okay? So, when you understand the group translocation, this is a very good slide there. First of all, you need to know where is phosphoenolpyruvates. It's right in the last step of glycolysis. It's an important step. This will generate the pyruvates, and the pyruvates further go TCA cycle or go fermentation, and this guy here also generates ATP. So that's an important chemical, and this chemical repeatedly used, then by the bacteria going through different groups, then they transfer phosphates directly become glucose 6-phosphate. And that is a very good slide, and this slides we will be mentioned again in later on when we talk about the lactose operant. We will mention that again. So that slide is important for you to understand. It's the most widely used transmission methods for the nutrients for the bacteria. Okay, so that's group translocation. Okay, next one, we want to talk about the bacterial cell wall. We already talked about in the Gram-Stain. Using Gram-Stain methods, we could differentiate the bacteria into two groups, gram-positive and gram-negative. So gram-positive stains purple, gram-negative stains red. And we mentioned about the two theory, which is cell wall theory, because gram-positive bacteria has a peptidoglycan, which is thick, and gram-negative is very thin. And we also talk about the lipid theory, because gram-negative bacteria have lipopolysaccharides. So this is, we're going to go down to a little bit detail in the cell wall function. Protect the cell from osmotic lysis, toxic material, pathogenesis. We're going to go to this slide to talk about that. Okay, so we're going to talk about cell wall structure a little bit detail, so for you to can be understand well. So we're going to remove these. So first of all, we already draw this. Okay. We use gram-positive bacteria as an example. We already draw this Royd mosaic model. Just draw it briefly, since we already talked. We draw it briefly. So we have these transmembrane protein. Let's just draw this. Okay, just draw this. And we also have oligosaccharides like here. Just say like that. If this is a gram-positive bacteria, you will see a very heavy area here. Now, of course there's space, okay? That's called Paris, that is called periplasmic space. Periplasmic space. This imatic space, we don't really care about too much, because that's an enzyme release or accept the material there. But what we care about is this huge area. This huge area, we call it peptidoglycan. Okay, we want to talk about this guy. So, peptidoglycan. First of all, very simple understanding. Peptidoglycan is composed by, if you just don't know anything, we just want to make sure it's easy, we can say peptidoglycan is composed by modified sugar cross-linked with amino acids. That's a very simple sentence. It's even a layman language. Now what's modified sugar? For a bacterial cell, so widely studied, two major modified sugar in peptidoglycan is NAG and acetylglucosamine and NAM and acetylmuramic acids. Okay, what are they? Why we say it's a modified sugar? So this is going to be a little bit of chemistry. Glucose. What is NAG? Okay, NAG looks like. You should know glucose. Okay. No matter what you get in your organic chemistry, no matter how horrible the class is, you need to know the glucose. Otherwise, you get big trouble. Okay, glucose like that. That's I told all my graduate students. there's any type of the information, but this is important, a glucose. Okay, this is the glucose. This is carbon number 1, carbon number 2, carbon number 3, 4, 5, and 6. What is N-acetylglucosamine? When you already see the glucose there, glucosamine, then certainly something going to add it. What are going to add it right here? NH2, C, and CH3. That's glucosamine. Okay, what is N-acetyluramic acid? That is based on NAG. You're going to add something. How are you going to add it? This carbon number 3 goes here. So you have a C, CH3. So you go here, you have a COOH. And this is usually CH, okay, like that. Now, this guy is important because this OH could be gone. If you carry on amino acids, go here. Is that right? If you go here, could go long and long. So that's N-acetyluramic acids. Now, how the bacteria is connected with each other? If it's gram-positive bacteria, they are connected like NAG, NAM, NAG, like that. And then there are some of the amino acids go here. Okay, amino acids 2, amino acids 3, amino acids 4. And then they go another one. Okay, right down here, so that could be NAM, NAG, then go NAM, like this. So you go up. Let's say here you have AA1, AA2, AA3, or AA4, they connect with each other. Now, why this guy is heavy? The reason is they are connected with each other, not like just separated. There are cross-link bridge here. Basically, is glycine. The glycine is connected. But for gram-negative bacteria, why this thing? They do not have this glycine cross-bridge link. And that's why it is very narrow. Okay? So what is glycine? This is a basic amino acid group. Is that right? Amino group, carboxy group, H and R. If this R is H, that's glycine. If this R is CH3, that is alanine. So that's going to talk in the exam 3, okay? That's the reason it will be very heavy. And once it's heavy, it's hold on peptide, hold on crystal violet. That's why I show the purple color. So you can look at the slides here, which tells you the primary structure of the peptidoglycan. It is interesting to see the cross-linked with each other. And this is a little bit of chemistry behind it. And then you go here, certainly very simple. Plasma membrane, periplasmic space. That's for extracting some of the, or release some of the enzyme. So let's form more activated. You have a cell wall peptidoglycan. It's very like thick. Then on the top, you have lipoteichoic acids. And this here, you also have a teichoic acids. And this function, lots of the textbooks said, I don't know, we don't know about that. Now, another thing I also want to mention, this peptidoglycan, here we talk about is a Gram-Stain principle, and you already know. In the exams too, we will talk about antibiotics. This is the one is they're attacking, because they have these links with each other. They have these links here, they have the link here. these will be a very vulnerable place for antibiotics to attack. So this tells you something. Why we have a very effective antibiotics? Because we studied the cell wall structure of bacteria very well. It's well recognized and well known, especially for gram-positive bacteria. But why we don't know, we don't have a good drugs for, or antibiotics, whatever you call for virus, because the virus structure is way too simple. It's not that complicated. The more we know the chemical behind it, the more we know the targets. We can create those drugs. But if we don't know the detail of that, we cannot figure them. That's why sometimes you know a little bit of detail of the chemistry, it's not a bad thing. It's a really good thing for you to understand the principles behind it. So antibiotics mechanism regarding here, we're going to talk later on. So just give you some of the hints, okay? Gram-negative bacteria, you could see a completely difference. There is three major differences, I would say. First of all, their peptidoglycan is very thin. And we already mentioned why it's very thin, because the cross-linkage of the amino acids is very short, or don't even have amino acids. Second, they have a double layer of the cell membrane, which is plasma membrane and outer membrane. The last thing I always say it's very important is this lipopolysaccharides. This is the LPS, on the outside. Okay, this is by lipids A, polysaccharides, and that's lipids A is a major components. This becomes O antigen and the core area. And the O antigen is important. That's what O157 comes from. You do a serological typing, you give them ID, you get O157H7. Now, what is H means? This is flagella. We'll talk real quick. So that's something we want to talk about. The gram-negative bacteria, lipids A, O polysaccharides, it is important. And this is another slide which gives you a clearly difference between gram-positive and gram-negative bacteria. And you can see them, compare these, more obvious, the difference. Capsule, just be very briefly, because we already talked about that. Capsule is polysaccharides outside of the cell membrane system, cell wall system. It's antifungal, resistant to the phagocytosis, protection from the desiccation, and exclude the virus and detergents. The picture here is Klebsiella ammonium. Klebsiella ammonium has a very heavy capsule. So, Klebsiella ammonium, when they grow on the agar, looks like a cheese. We call it a milk-coiled colony. We will mention real quick. It's called a cheese-like milk-coiled colony. Very sticky because of the heavy capsule. And we could do capsule stain to identify it. And we already talked about the capsule stain. It's basically we can do negative stain, or we can use in carbon sulfides to do directly staining the capsule. Okay? Ribosome. This one I want to mention a little bit because people sometimes will get confusing from here. Okay, ribosome, first thing first. This is a few of the machinery, both cell machinery, both prokaryotes and the eukaryotes they have. That's a ribosome. And the ribosome is for production of protein. And they have two subunits. So basically it looks like this. For bacteria, this is 50S, this is 30S. However, add up together, 70S. It's not the mass. Is that right? 50 plus 30 should be 80, but why this is 70? This the Van Berg unit. That is to testing the speed of precipitation. Okay, it's not a molecular weight. So when you add up together, it's 70S. The big subunits, we have 23S and 5S subunits. But the more important, at the 30S subunits, we have 16S. And the 16S RNA could recognize a